Hello. So good morning to everybody. It is a pleasure that you are here, and it's even more a pleasure for me to introduce you our speaker, Professor Maria MINUNNI. She has an international renowned expertise in aptamers mobilization and chemical biomedical sensing applications. She is chair of analytical chemistry at Dipartimento di Chimica Ugo Schiff, Università degli Studi di Florenza. She worked at the different companies, Nestlé Research Center in Lausanne, at Pharmacia Biosensors in Uppsala, at the new Biocore Instrumentation also, at the Technical University of Munich, and at the University College of Cork. Afterwards, she returned to Florence, where she works in the Department of Pharmaceutical Science, and then she went to the Department of Chemistry. Her activity has been mainly focused on the development of bioanalytical assays, mainly on sensors and biosensing-based approaches. In 2000, she returned to Florence University, and she became a associate professor in 2011. Afterwards, in 2017, she achieved the full professor position at the analytical chemistry area. She supervised more than 20 Master of Science and 5 PhD students, and most of them are now developing academic careers, not only in Italy, but also abroad. She has co-authored more than 1,025 papers and reviews articles in high-impact journals. She has also, and she is currently a principal investigator and coordinator of different national and international projects, mainly in the field of piezoelectrical and optical biosensors. And I think that now it's time to allow to Maria to start her presentation. So thank you very much for being here, Maria. It's your time now.
Thank you very much to you for the kind introduction and invitation. I really enjoy it. At this time, it's very nice to keep in contact somehow, even in remote. And I will start with let's say describing what are the main characteristics of a sensor, let's say optimal one. So we can imagine a minimal sensor pretreatment. Think to the glucose sensor for glycemia, which is an electrochemical device. Patients can do it on their own at home. It was really a revolution when it came to the market. You can modulate the specificity, and the specificity, as we'll see, depends on the immobilized ligand. You can automatize the data acquisition. It's non-invasive, eventually in case of ceruloplasmoresis, for example, piezo label-free technology. And this has the advantage to reduce the labeling procedures, reducing costs of an analysis. You can have, for example, affinity and kinetic measurements, which are very useful eventually in bioanalytical things, as well as in drug development. You can calculate the kinetic cost of an interaction. As well, you can have also qualitative and quantitative estimation. You can have a rapid analysis. Not always is necessary to reach the equilibrium of the interaction. And if you couple to sampling system, auto-sampling, flow injection on eventually online configuration, you can have a high sample throughput. And another advantage, since bioreagents are very expensive, is the possibility to use low reagent consumption and eventually through miniaturization of the sensor. And as well, as I said, for the glucose, we know since years, you can have a minimal demand on user time or skills by automated analysis. Let's see. Yes. We can distinguish between two classes of biosensors, like catalytic, where the biological element, generally enzymes or enzyme systems, convert the substrate into product, and you can monitor either the product formation or the depletion of the substrate. And you can have affinity sensing, where the biological element, called receptors, binds specifically the analyte leading to a complex that you have analyte binder and you have the complex AB, and this can be reversible. So the transducer in this case reveals the complex. What we do as analytical chemists, which is our approach. So now let's consider for a while which are the variables that we should consider when designing a biosensor. Normally, we don't consider as chemists the signal transducer processor. You are the best expert for this. But we concentrated best on the analytical problem, which means which is the analyte I want to detect in which matrix, and that means which is the concentration level I have to face. Then, which is the biological receptor suitable for this approach? Which kind of interaction between DNA, nucleic acids, protein, whatever. And the third strategic thing is the mobilization chemistry, how I want to mobilize to bind the receptor to the transducers through adsorption, polymer entrapping, covalent binding. We generally use covalent binding because we use delicate surfaces, quite expensive, but you can always use absorption if for a single-use sensor, for example, can be okay, not always, but. And in definition of analytical problem, we have to face the kind and of the analyte and the level of the analyte. Now, for the application, very popular application to molecular diagnostic, we have the need to push the research towards ultra-sensitive detection of biomarkers, and together with the easy accessible matrices. And this is the key issue to apply these devices to anticipated diagnostic prognosis and therapy. And eventually, let's focus on the healthcare problems. Which are the challenges in this area? Well, diabetes is the fastest growing chronic disease in the world. Heart disease and stroke kills tens of people per year. It is 1/3 of the whole deaths globally. Cancer, of course, and aging population, neurodegenerative problems, for example, the demographic time bomb, we will have an increase in this part of the population. So to face this, we have to concentrate on sampling. Analytical chemists cares a lot of sampling. And the question is what to search and where to search, in which matrices. So let's have a look what is done routinely. Normally, you have, for example, solid biopsy, which is the gold standard, and there's invasive, costly, and risky, but you have to do it if you have a problem, a serious problem. And you maybe need to be anesthetized, and can be painful, of course. And there is a problem sometimes with this kind of sampling because some tumors are not homogeneous. So when you sample, you have to be sure that you don't have false negative, and you are sampling exactly the tumor. And basically, you have 1 million people that, for example, do breast biopsy only in the USA. On the other side, there is the liquid biopsy, which is taking place as a non-invasive, cheap, and non-risky, and it faces better the heterogeneity of the tissue around sampling, because you have a matrix where all the molecules are there. You have to pay attention to small things, but it's much better to face the heterogeneity of the tumor, and eventually you can perform several sampling over time to look, for example, the changes in the markers, and eventually to relate them to the response to the chemical treatment, like chemotherapy, for example. These matrices are very complicated. You have a whole blood here, you have cells, which are lymphocyte, white blood cells. We will talk in a minute. You have erythrocytes, no nucleus, but a lot of hemoglobin. And you can separate them, get the plasma, and you can get eventually the serum, which are the most used matrices. And the difference is that the serum does not contain fibrinogen, which is the molecules involved in the clotting cascade, because in serum the clot is separate, while is not in the plasma, which contain fibrinogen. So if you have to look for this molecule, fibrinogen, you have better to look plasma. Otherwise, in serum, of course, you won't find it. And in blood, you can, for example, in cancer screening, you have some possibility. You can look to circulating tumor cells or to nucleic acid, for example, and or to exosome, which are new emerging very important structures, vesicles containing proteins and nucleic acid, DNA, RNA, which are used among cells to communicate. So these are involved also in metastatic problem of the cancer. So it's an emerging, let's say, marker structure. And look at DNA, for example, you can look at point mutation, which are very important to decide the chemotherapy, for example, to be delivered to the patients. And again, you can have a urine, even easy to sample. And also, if you have a blood vessel here, and you collect information from all the organs, and the biomarkers are there, then the blood is filtrated by the kidney through glomerular filtration, and some information passes through the urine as well. So you can have also urinary cell-free DNA, and eventually you can collect also information from bladder cancer or prostate cancer for men. At the bottom you have a urine sediment. In the supernatural you have this kind of. You can have, of course, proteins in case of kidney failure. This is also very important. Albuminuria and other measurements about proteins in urine. And as we know, we have seen many times this here. You can have an naropharyngeal swab and saliva, which are also sampling very easy to sample matrices and very useful in diagnostic. And finally, we'll see an example about the cerebrovascular matrices, which is not so easy to access. It's quite, it's a bit painful, but it's very informative about central nervous system problems. And so you, in a vial, basically, with a liquid biopsy, you can have a sort of panorama about the state of the human being. But what we have to face, of course, in liquid biopsy, you have a low level of analytes in these matrices. So the problem is the sensitivity of the device. So for this reason, you have to face ultra-sensitive detection, both of nucleic acids, biomarkers, all this we have seen. microRNA as well is important biomarker in cancer, and protein biomarkers. And you can do that by natural or biomimetic receptors. And we will see a few examples of this. Which is the strategy to improve the biosense analytical performance in terms of sensitivity and reproducibility? And this is very important to work in real matrices. We know of very little sample pre-treatment. Otherwise, you lose the advantage of working with sensor if you have a longer sample pre-treatment. The one it's useful can be just a dilution in case. So the ways to work in this direction to improve these two parameters is, let's say here schematized very briefly, it's improving the analytical signal. Well, better standardization of signal sampling and management. Point two, standardization, optimization of the receptor, how to select the right probe, eventually coupling the system to nanotechnology for enhanced sensitivity, and decide a suitable surface chemistry to avoid or reduce a specific binding. We will not concentrate on this because we have a time reason, but I will go deeper in the first three in case it's open for discussion. Of course, as anticipated, we work with label-free technology and eventually with certain plasma resonance. So basically, I will not explain it, of course, you know better than me this, But basically, the technique monitor changes in refractive index that reflects on the changes in the resonant angle. So basically you can monitor the events that occurs at the interface by looking at the change in the resonant angle over time, which is in the commercial instrumentation referred as resonance unit changes signal. And this is what is called a sensorgram, the displacement in real time of these changes. And if you have an event here, this changes the binding event concentrate basically the molecule, the interface, so the local resonance refractive index changes. So you see that through the signals. So which kind of information you can you can have? You can have information about quantitative, so active concentration, how fast, about kinetics, about affinity, and about specificity. So what is done is this: you immobilize a molecule, a receptor on the gold surface, which is eventually chemical modified. You inject your analyte. If it binds, you look at the association of this interaction, the formation of the complex. Then at a certain point, you stop to inject the analyte, you wash the surface, and you see the pure dissociation event. And you can dissociate this complex using by low pH, the chaotropic agents, increasing the ionic strength of the solution, and then you can come back to the baseline. It's not always obvious to come back exactly to the baseline. And this is very important, very, very, very important, because if you don't come back to the same value, you have a different surface capacity at a subsequent cycle. And this affects very much the reproducibility of the measurement. So one person has really to care about dissociation optimization with the selecting the solution, the time, and things like that. So this is, we have seen the SPR with one or few sensing area, which is the most of the instrumentation, commercially available instrumentation, and where the detector are array of light sensitive diodes. But the evolution of this instrumentation, the principle is the same, went through SPR imaging, where actually you have a lot of sensing area called the region of interest, and eventually up to hundreds, and you can monitor simultaneously many analytes, and you can eventually by a CCD camera looking at the spots where you have the receptor. And this is very convenient, for example, if you have an air bubble, and eventually to evaluate the distribution of your receptors. We'll see in a moment on the spot. And you see, you can there is a scale of color. No binding is the blue, and binding the maximum of the binding is the red. So here is intermediate, let's say low binding, for example, just to give an idea of the instrumentation we have used in the last years. And here is the chip. You can spot a solution on your chip. We prepare normally some polymer, dimethyl siloxane layer. It's a polymer very useful, polymer in fluidic, macrofluidic as well. We pierce the polymer, we obtain some welds, micro welds, and then we modify the surface using the DNA probes mainly, with the thiol. The thiol has SH group, let's say, and which has a high affinity for the gold, so you can consider it mostly covalently bond probes. And then we passivate the surface with other thiol, short chain, mercapto on hexanol, for example, and with a mixture. It's important also to have like a brush surface, and this is very important to prevent non-specific adsorption, because any molecules sitting on the surface give a change in refractive index. So we don't want that anything sitting is taken as analytical signal, but we want to be selective that just sequences binding the probe are detected. So it's very important the chemistry you apply here. Then you can immobilize different probes, so with different sequences, as well as negative controlled surfaces, and you can go for the analysis of this. What you get actually is a CCD camera image, and then you can spot you can decide how to take this analytical signal. You can take one spot, or you can a subset of spot. You can generate a grid, as we suggested. And you see here, it's very evident that the receptor deposition is not uniform. You have a spot heterogeneity. Mainly, it's concentrated on the border, at least in our system. And you can detect how to take the signal here. Try to optimize it. And we have seen that depending on the sampling, signal sampling, you can improve the analytical performances, because if you select a rational choice, take, for example, the signal up to a certain level, eliminate things like that with a macro, Excel macro, for example, then you can differentiate on the basis of the same data. You can differentiate the gray bar, which present higher signal, but which is very important, is a standard deviation, the low standard deviation. This is one things that contribute to the positive analytical performances. Another thing is the receptor selection optimization. Imagine to have a gene sequence and then to go for some probe selection in silico, depending on some parameters, which is the melting temperature of the DNA, the folding structure, etc. And you can get some scores ex ante in silico. And what you could do, you can perform an experimental validation by optical sensing. Actually, we have seen in the past that this ex ante evaluation on in silico gives important information on the target, on the probe performances. Here is, for example, if you inject a certain amount concentration under the nanomolar, but which is here actually, but you have the same trend all over the concentration range tested. So the right evaluation of the probe sequences, it's very important to improve to push the analytical performances. Just to tell you something more, how you reach then the ultrasensitive human genomic detection up to attomolar detection, for example. And this is what I will show in a minute. You can take a gene, for example, which specify in ATP binding cassette transporter involved in pharmacokinetics alteration versus some drugs like digoxin, for example, and with interest in pharmacogenomics and molecular diagnostics. Nowadays, it's known that dose and medicine selection are based on the genomic profile. So if you have some mutation, for example, like in this gene, you will not go for a certain therapy, but eventually also the dose of the therapy is dependent on the genomic profile of the patients. So what we will see is that how we will detect a point mutation, a single nucleotide polymorphism in this region by DNA extracting from white blood cells, lymphocytes basically. To achieve this, you have to work in a SEI design. So you select the gene. We have seen this, which gene it is. You select the right probe, right in silico estimation, and then you try to think how to detect, to use this probe. For example, one probe can be used as capturing probe you mobilize on the surface, and the other we can call detecting probe. You can use to bind a second region different from the previous one, and eventually where the mutation is. In this slide, we see actually how you can use this capturing probe, and that is also very important to optimize the length of this probe and eventually some modification you can add to this probe, because if you have a mutation here, it's important to discriminate in the analysis if you have if you are a patient with no mutation, if you have the mutation, because on the basis of the mutation you have, you will get the treatment. So it's very important to distinguish between the full complementary binding and the mismatch. So which means one not fully complementary binding. So it's important to distinguish between a full stabilized hybridization and a very much destabilized interaction to distinguish better the situation of match and mismatch. And you can see that depending on the length of the probe and the chemical modification on one hand, you can amplify this difference. So the better situation is where you can, the full match gives a signal, and the mismatch compared to this gives the lowest signal. So you can say with a certain certainty that you have not the mutation, or you have, as in this case, the mutation. And if you apply this approach to white blood cell types, and you apply some sample treatment like ultrasound fragmentation to cut in pieces the length of the genomic DNA, which is very, very big, you reach the detection down to sub-femtomolar, which is quite a respectable concentration. Then you can eventually use plasmonic nanostructure with DNA, as we say, to enhance sensitivity. What we did was just to push further the sensitivity, the DNA sensor sensitivity, to use eventually to explore the effect of coupling between surface plasmons and electric field of localized sulfur plasmons of nanostructure of different origin. So basically, we added to the secondary probe, the capturing, the detecting probe, different nanostructure showing no plasma, like ACO2 nanoshells, silicon nanoshells, gold nanosphere, and silver nanoplates. The best one displaying good performances were the silver nanoplates. And you can see here the capturing probe interacting with the complementary, with the addition of the secondary target without any structure, and the different presence of the different nanostructure. You can see from the color itself that the interaction is higher, and these are the signal are the difference compared to the first one. So it's considered just a secondary probe, and you have an increase because this is explained because the plasma here is placed at 630 or 33 nanometers, while the laser here was around 600 nanometers. So it's explainable. And we further improved this approach using gold nanostar, because here you have the tip effect, which is a bit higher compared to the previous approach. And you can see that here we were able to reach attomolar, which is 10 to the minus 18 mole per liter of the target. This is of the target analyte using a femtomolar DNA, human genomic DNA. And what we can say, in this sense, we can say that with this system, you can use parallel gene detection, multi-gene detection. You can define the polymorphism profile used for drug therapy or drug resistance as well. You can have, in case of nanostar, you can reach attomolar detection with a selective and regenerable, this is a very important, sensor chip. And the effect on the nanoplasmons, of the nanostructure plasmon, particle shape, material size, impacts on the signal. And with this in mind, we can go for circulating free DNA. We have seen biomarker in liquid biopsy in the molecule we have seen in this kind of samples. And these results are in agreement with recent findings exploiting a liquid biopsy approach and using ultrasensitive gold nanoparticles for detecting at attomolar level oncogenes KRAS polymorphism in the plasma of colorectal patients. And using the same sandwich lac approach, and this was done within a consortium. This project is ended already, and we were part of this project like in this consortium, National Institute of Biostructure Biosystem. The coordination was in Catania by Professor Spoto, and the nanostructures, also the chip was a nanostructure here, and was Professor Romola at the Institute of Photonics and Electronics in Prague, who did the physics of the device, studied all this. And in literature, you can see which are the performance of the plasmonic platform for the detection of circulating biomarkers in biofluids. And you can see that you are at zeptomolar, femtomolar, and in plasma and serum, and again femtomolar for looking at the DNA level, RNA and other DNA level. And so we can say that this limit is reachable. Briefly, I want just to say that you can use this enhancement with nanostructure also in case of detection of proteins like tau protein, which is involved in Alzheimer's disease. Eventually, in liquid biopsy in the cerebral spinal fluids, simulated matrices, and this work was done in collaboration with Professor Rick Perrin at Grenoble Alpes University. And we have been working using antibodies as receptors, or as at Grenoble they developed aptamer for tau. And we work with the antibody using SPR and eventually piezoelectric sensors, again label-free. I want just to mention what happens when we use these nanostructures. So basically you have an antibody as a receptor, you have your target molecule, you have your shift. The target changes the shift, and eventually if you use a sandwich assay with the nanostructure del multi-wallet carbon nanotube, you have a big shift in the resonance, because the refractive index of these tubes is really high. And what you have to be careful is to optimize, okay, because the nanotubes population is wide, to select the small size carbon nanotubes, otherwise you have a mass on your chip. And also in this case, you can reach picomolar level with a few minutes of analysis time, and good analytical results in buffer simulated molecules, because we didn't have no patients in this case. And this is just to show how, if you have an unlabeled secondary antibody, and if you have the label with the nanotubes. So, of course, the contrast is batch-to-batch variability, eventually a decay generation problems. And just to mention localized phosphate plasma here, test appear over years biosensitive with plasmonic nanosensor, and the differences between surface and localized plasma resonance is that the first one is sensitive to bulk effect, let's say. It sample an area longer of the vanishing way here. And while localized plasma is confined in a short length depth. So basically, you can obtain, if you have here two kind of responses, you can have a better ratio between the specific antibody response and the buffer response. So this improves sensitivity. So many people are working on this. And you can nanostructure the surface, so you can have a plasmonic and nanoplasmonic in solution on surfaces. And the spectra you record depends on the shape of the material, as you know. So LSPR is applied to affinity biosensor, homogeneous in solution, or with a nanoparticle immobilized on the surface, and you can look at the shift on the plasmon. There are some commercially available instrumentation. These are the chip, nanostructured chip. And the advantage is that you can, in this case, actually have a better, higher instrumental simplicity. No adapted optics, or you can also have low cost in the development of instrumentation and chip preparation. In the last minutes, I just want to talk about briefly the possibility to use polymers and nanostructure together, and to how to build up biomimetic receptors. So how you can develop a nanocomposite for plasmonics, and how you can use nanostructure for ultra-fast polymerized photonic PCR with ultra-fast polymerase chain reaction to amplify nucleic acids, and eventually the use of new molecules which are present, the catecholamine in human body, to develop new receptors. So starting from, for example, polydimethylsilocene, you can do a lot of things. It's an interesting polymers. It's transparent, so can be used in optics. You can do microfluidics, you can do micro nano machining, and also you can prepare microneedles for blood sampling, for example. We approached recently the possibility to develop chip on PDMS by synthesizing gold nanostructure on the top of the PDMS, and to explore them using a portable spectrometer. So you can, you can modulate the spectrum, change the composition of the gold, or the solution to obtain gold. And then you can change, for example, here, you see, and then this is wet, this is dry. You can sample the different spot and look at their spectrum, which is wide depending on how they were prepared. And this is one thing we started. Of course, we are not expert in this, but this is a possibility to produce simple and cheap devices to be miniaturized eventually. And another thing is the ultra-fast photonic PCR based on photothermal nanomaterials. And the advantage of this, people say that in PCR, conventional PCR, you have a too long, prolonged reaction times, while the photonics significantly shorten the PCR reaction time, and that shift next PC breakthrough after quantitative real-time PCR. So it's a revolutionary thing. And the advantage is that photonic PCR features volumetric heating and non-contact heating, which minimize the energy the time consumed in heat transfer and expand the application of nucleic acid amplification. So in principle, you can have cheaper, portable, and faster PCR devices. There are some review of this, and you can use different nanostructures. So you irradiate with a laser, and then you can the heat diffuse to the solution, and you can increase the rate of the PCR in this way. There are different nanomaterials for ultra-fast bead-reported PCR with different performances, and last day has been reported that Korea Advanced Institute of Science and Technology, KAIST, developed ultra-fast real-time photonic PCR for detecting COVID-19 in just 5 minutes. Okay, this is, let's say, a journal news, but this means, the past. We are not at this, but we had the project under photonic sensing and where we were able to demonstrate how could work the photonic PCR if that functionalized probes, primers, do not impact on the PCR, because it's a nanostructure that goes into a reaction, so you don't know how it works. And we couple that to the SPR sending. So amplified PCR photonic can be used also in nanosensor. And in 2 minutes, I'll just go to the last things very quickly. And again, with polymers, inspired from nature, you can do a lot of nice things. There is a molecule which is called a polymer starting from dopamine, called polydopamine, and there is an analog of polynorepinephrine. Polydopamine is present in the adhesive plaque in the pedunculus muscle foot proteins, which sticks on the rocks of the muscles of the sea, and norepinephrine does the same with the possibility to have a smoother surface at the nanometer scales and enhance hydrophobicity of the functionalized surfaces, as you can see, smoother surfaces, and the contact angle is quite different between these two. This is untreated surface, polydopamine treated surface, and other surfaces. The paper on the polydopamine research has increased over time significantly. You can do a lot of things. What we do? One thing is that we are working to develop biomimetic receptors alternative to antibodies. So if you take a monomer of dopamine, for example, and you let it polymerize in presence of the analyte, and then you wash off the analyte, and the polymers present some cavities that fits with the analyte. This you can do this, inject in the rabbit and get the antibodies. So,
Maria, you have 1 minute.
Okay, you can have the advantages of MIPS compared to antibodies, fast design, low cost, and these are small molecules, hormones basically. You can functionalize your surfaces, either the chip or polystyrene, and you can get some surfaces, artificial surfaces, you can use for modifying the surfaces for peptides. For example, you can select some part of peptide and develop portable sensors for a plethora of analyte. We have seen cardiac markers, peptide hormones. This has been of interest in anti-doping analysis in urine. And you can do a lot of things for the different applications. And after chip, you can go to cheaper systems. And this is, let's say, the end. This is the future. Where are we going? So, plasmonic biosensor for single molecule biomedical detection. There is this review, very recent, which gives you the panorama about the concentration span between and what you can detect in this range. The conclusion, I hope I demonstrated that the analytic approach can be useful to push forward the limits of these systems, the role of nanostructures, and the role of chemistry, and what you can do with this biomimetic receptor as synthetic antibody for protein peptides, which decrease a lot the simplicity and the cost of the analysis. And I want to like to thank again you for the attention, and the committee for the very kind invitation. Thank you very much. I'm open for the question.
Thank you, Maria. It was really very interesting just to show us, how important the interdisciplinary is, because, you have a huge knowledge about things that are completely complementary to most of the audience. I'm afraid that, we are just out of time, so we cannot have the questions now. But I have been talking to the organizers because I know there are a few questions, and they are going to send it to you. So then you can just directly, if it's okay with you, just to answer them. Okay.
Yeah, sure. I'll be more than happy. Okay, I'm sorry I took all the minutes. I thought it was time, but the last I didn't look at the club.
But I mean, it was very interesting. So I think everybody appreciated that you keep on going, just explaining everything. But like I said, we can leave the questions for afterwards. Okay. So thank you very much.
Yeah.
Okay. Bye-bye.